Assalamu alaikum students. Today's lecture is about meiosis. Meiosis is actually a type of cell division in which gametes are produced. Gametes may be either male gamete or a female gamete. They are produced via meiosis. Meiosis is actually a process in which diploid parent cell is converted into 4 haploid daughter cells, which means that the number of chromosomes that are present in the parent cell is halved in case of daughter cells, which helps during sexual reproduction. There are 2 sets of cell division which are involved in meiosis, meiosis 1 and meiosis 2. Meiosis 1 is further divided into prophase 1, metaphase 1, anaphase 1 and telophase 1, while meiosis 2 is also divided into prophase 2, metaphase 2, anaphase 2 and telophase 2. Meiosis 1 is very much different from meiosis 2. Meiosis 2 takes place just like mitosis, which we learned in the previous lectures. So here we will discuss only meiosis 1. If we talk about the prophase of meiosis 1, then it is the longest and complex phase of meiosis, in which the chromosomes condense, synapses occurs, tetrads are formed, and a very crucial and important step, that is crossing over, takes place. Firstly, we will talk about prophase, in which the chromatin material is condensed in the first step, which means that firstly it was present in the form of thread-like structure and then it condenses to form chromosomes. Firstly, the process occurs which is called synapses, which is actually the pairing up of chromosomes by which the bivalent chromosomes are converted into tetrads. Now they have 4 sister chromatids, that is why they are called tetrad, and the process by which tetrads are formed or pairing up of chromosomes is formed, it is called as synapses. The points of attachment of the 2 chromosomes is known as chiasmata. Next, the process which is crossing over takes place, and the 2 chromosomes exchange their parts from the part from which they are attached to one another, and they exchange their genes, which results in the form of new varieties in the next generations. The second step of meiosis 1 is metaphase 1, in which the chromosomes that are in the form of pairs are aligned to the equatorial of the cell and form a plate known as metaphase plate. The centrioles which are present at the edges of the cell start producing the spindle fibers, and these tetrads get attached on the equatorial plate on these spindle fibers, and this metaphase plate is produced. The next step of meiosis 2 is anaphase 1, in which the chromosomes are pulled towards the poles by the help of these spindle fibers, and half of the chromosomes in the form of bivalent chromosome is pulled towards each pole. The next phase is telophase 1, in which these chromosomes are pulled to the opposite poles, and then the nuclear membrane starts bringing up or building up around them. Then another process which is called cytokinesis takes place, by which distributes the cytoplasm between 2 cells, and thus the 2 cells or 2 daughter nuclei are formed at the end of meiosis, and these have the same number of chromosomes as the parent cell, as you can see, and some of the fragments of the chromosomes are also exchanged, which will result in the new variations or new characteristics in the next generation. At the end of meiosis 1, prophase 1, metaphase 1, anaphase 1 and telophase 1, 2 cells are produced from a single parent cell, and the number of chromosomes are same as they were in parent cells. And then meiosis 2 takes place, which is similar to mitosis 1. During meiosis 2, the 2 daughter cells that were produced in meiosis 1, they are converted into 4 haploid daughter cells, and they have half number of chromosomes as compared to the parent cell, which is involved in the sexual reproduction of living organisms, and which gives due to crossing over, it gives the new variations and new characteristics to the new generation. Thank you.